Thank you very much, and thank you for the invitation to be here. And I took the task quite literally, so I'm really going to talk about applications focusing on MEG, but of course it would apply to EEG and frankly ACOG as well. So the style of the presentation is a little different from what we've heard so far, but I really want to get into thinking about the features of electrophysiology. We've heard a lot about that, what we have in the time domain, what we have in the frequency domain, and how we can harness those features to give us something application-wise. So every now and then, up is going to pop a clinical application, but always based on some property in the temporal or spectro-temporal domain. I'll say some disclosures. And generally, the role that we are seeing for electrophysiology, the emerging role, is the pursuit of biomarkers for some disorder. That is a non-invasive, biologically based indicator or signature that has a variety of uses that may be diagnostic or may be something to do with clinical trial design, inclusion criteria, measuring responses to therapy. And we specifically are very keen that there's a biological basis to these things, that they're not a bunch of electrophysiological epiphenomena. And so throughout this presentation, I'm going to step outside of electrophysiology and just say, can we get supporting evidence from something else to help us with our interpretations? So these are the three machines I'm going to show data from. These are different MEG machines that we have in Philadelphia. They, broadly speaking, have the same principles that we've heard about last month. Many detectors over the surface of the head, non-invasive, no surgery, no electrodes actually sticking to the head. The cute thing is the one on the bottom left. That's a very small one. That's kind of like a mini meg for babies, and that's primarily because of R-squared. Magnetic fields falling off with distance in a really unpleasant way. They weren't strong to begin with, and then they get very weak very fast. So you have to get very close in order to be sensitive to them. So I'm going to contend that MEG, and to a lesser extent EEG, and to a sampling extent not at all ACOG, is really something we can think of in 5 dimensions, since we have 3 dimensions of space, a 4th dimension of time, and notwithstanding the Fourier transform that relates time to frequency, and notwithstanding the perils associated with binning the broad frequency domain into traditional bands, since different things are occurring at different frequencies, whether they're different processes or different types of process, we can think of there being some extra information that's embodied in that spectral content. So forgive me for the mathematical stretch, but let's think of it as where in the brain, when in the brain, and then what in the brain. And again, emphasizing in the brain, although we don't go in surgically with subdural grid electrodes, we do mathematically get ourselves into the brain to elevate the field from the previous history of sort of scalp-based recordings and sensor-based measurements. We don't actually care about sensors, we care about brain. And so we have tried mathematical manipulations, and again, this was covered a lot last month. There are a variety of ways of getting the signals from outside of the brain and interpreting them inside the brain. And we're going to talk about how to do that. So it's sometimes called going to source space or getting into the brain. And the reasons that we do that are threefold. The first one, I think, is very obvious clinically, is so we can have an anatomic interpretation of the source of the signal. That's sort of, I think, the one we first think of. Where is the motor cortex? It matters anatomically that you get that right, especially if you're planning surgery. So that's important, but it's not the only reason that we try and go into source space. So another couple of reasons that I think are really under-discussed are that you get a signal-to-noise increase, because effectively you're signal averaging, because you've got many sensors recording, and you're going to somehow combine those to give you a single brain time course, a brain activity time course. It's not a root N type signal averaging, but it's not a bad form of at least leveraging the information at multiple sensors and combining it. So you can think of that as giving you some sort of signal to noise increase. And the other thing is that by going spatially into the brain, we can reject sources that come from spatially other areas. That might be other areas of the brain that we don't care about, or more likely it's the eyes and the muscles of the scalp, and frankly even the heart, and other places that are contributing to our sensor level signals that we simply are not interested in these experiments. And so we're able to project away spatially distinct artifacts when we go into source space. So,
what was that last phrase? You're able to project away what?
so spatially resolved. So it's a very simple concept. If you have a signal coming from the eyes, some eye blink or some eye movement, when you consider a spatial model of where the signals are going to come from, it's going to come from the eyes. And so you could say, well, now I'm not going to look at the eyes, I just want to look at the brain. So you can spatially filter the data, I think. The point is that the first one, which is the anatomic interpretation, is what you think of first. But these other two features are valuable to us as well, especially in contrast to scalp EEG, where we are often contaminated by muscle artifact or EMG signals that we sometimes mistake for Gammaband activity, and sometimes we mistake for broadband noise, and actually it's muscle, and we're typically unable to spatially project that away. So it's an important advantage of the source-localized approaches over the sensor-based approaches. The reason I want to, and this is one way to do that, and I certainly don't want to do this. I just was worried that everyone else was going to show a lot of math, and so I wanted to show a little bit. But this is at least one way to get into the brain, is this idea of the beamformer, or the radar array, if you like, that allows you to construct a time course of brain activity signal in the brain based on the x of t that you measure. And how you get that, the variety of assumptions that are built into that, but you can essentially create an electrode that you can stick anywhere you like in the brain virtually without having to actually do surgery. So let's think about the properties that we might want to look at in these brain signals. And I'm going to argue that there's timing properties and there's spectral properties, and things that we might be interested in that are resolved in the time domain are things like latency, just the time that things happen, the peak. And then more, let's say, advanced analyses of time domain signals. One that we popularly use is called excess kurtosis, so the fourth moment. That's a spike detector. It's used in epilepsy research and, frankly, in epilepsy practice. We might also compute the complexity, which can be formulated a variety of ways, but commonly is done in terms of some form of entropy measure, multi-scale entropy perhaps. These are all features of the time domain, and I'm going to show you how they're useful. Here's just a simple example of the spike detector, so that we're able to put a virtual electrode everywhere in the brain and get a time signal from every point of the brain, and then say, tell me about your excess kurtosis. Don't tell me about your latencies, don't tell me about anything else, don't tell me about your power, just tell me about how spiky your signals are. And then show me only those voxels that are very spiky, and then let's go back to neurology and determine whether those are clinically meaningful deviant spiky behaviors in the time domain. So that's the time domain from a particular voxel. Other voxels don't show that spiky behavior. And so that immediately has a clinical application in identifying the source of a seizure disorder like epilepsy. So that's how the time domain information can be useful. Let's think about what you can get in the spectrum. Of course, you can get the amplitude of a given band or of a given frequency or whatever. You can look at changes in the power up as populations, the rhythms get synchronized. You can look at changes in the power down as neurons get desynchronized, ready to do something else. So those are popular formulations. ERS and ERD basically mean band-limited changes in power associated with an event. And of course, like every other spectral thing, we have phase and we have frequency. And to the extent that more complicated things are going on, we can actually start looking at the interactions of different frequencies. And commonly, this is done by cross-frequency coupling. I think we heard earlier about theta, slow theta rhythms driving higher frequency gamma oscillations. So the interaction between these activities in different frequencies is in itself insightful. So here's that breaking it down into different bands. That's essentially saying you can decompose a broadband signal into a slower activity and faster activity. And the first thing you might do for that is to say, well, I only want to look at slow activity, or I only want to look at fast activity. And another thing that jumps out at you straight away, and this is historic data, this has been around now for 20 years, is the idea that if you do that, sometimes you're able to see slow activity marked in red in the signals, which could be on the EEG or more likely on the MEG. And the frequency of that, you can look in the spectrum and notice that the frequency is at about 4 hertz, the peak of the spectrum there. That's pathologic. Healthy adults don't have 4 hertz brain activity. You see this activity quite commonly around a tumor, quite commonly associated with seizure disorders. And in the absence of those conditions, you see it quite commonly in mild traumatic brain injury. So it's yes or no in adults. You don't have this in healthy adults. You do have it associated with trauma. So simple-minded spectral analysis of just saying, is there any power in this low-frequency band, is in fact being used essentially diagnostically for traumatic brain injury. No fancy stuff here, but it's something that's free from considering things in the spectral domain. Now let's take it a little bit harder than just saying, what about the power distribution? So this is what I was talking about before, about things associated with an event. Synchronization or desynchronization of a neuronal population associated with doing a task. And the reason that we like to do this is that whilst simple sensory experiments like flashing a light and playing a sound and tapping on a finger, those generate very nice ERPs, averageable up stimulus-locked events. More cognitive things, more complicated things, things where you can't really guarantee that the brain is going to do it on time every single time you do it, like remember something. Now remember it again, remember it again. It's very difficult to do that in a stimulus-locked way. We're able to look over a broader period of time at changes in power of some broadish band of oscillations, either in the alpha band or in this case in the beta band. So somewhere around 15 to 25 hertz, we look at the ambient power, some amplitude of this oscillation, and then execute some task like present a stimulus, and wait a little bit, and observe in a window afterwards whether the power has changed downwards, because desynchronization of the neural population is what is happening while those neurons go on to do the processing that they want to do, in this case, interpretation of a word stimulus prior to making a response. The point here is that we can do this for every single electrode that we virtually stick into the brain. So that is, every single voxel of brain can be interrogated with the simple question, do you exhibit a change in power when you see a stimulus? And more specifically, a couple of 100 milliseconds after you see a stimulus. And we can ask that question with a t-test, correct for multiple comparisons and all of that sort of stuff. But basically, we could say, only show me those voxels in the brain that do show this significant amount of event-related desynchronization. Suddenly, we have a map, a functional map of language processing based on neural activity. So every single voxel is then characterized, and of course can't show all of this dimension, but every single voxel, and I've painted one in green there, is characterized by a time course, that's the x-axis. Stimulus comes on at time 0. And it is characterized by a frequency domain, a spectral, like a power spectrum of activity. And the stimulus comes on here, and desynchronization, so a drop in power will be shown as in this case, as blue on this color scale. So you notice that yellow means 0, so there's not much going on in baseline in this voxel. The stimulus comes on, 200 milliseconds of not much happens, and then suddenly we see a blue blob. And the blue blob here is specifically saying that after seeing a visual word, this superior temporal gyrus voxel exhibits, or the neurons within that demonstrate alpha and beta band desynchronization that that lasts about 500 milliseconds. So we're now able to say with some specificity when things are happening, where things are happening, and actually what is happening. In this case, alpha and beta-ish desynchronization. And of course, you can do that as a way of mapping function. So this is another clinical application, pre-surgical mapping. In this case, not of a language task. This is looking at the motor or sensory motor activity associated with pressing a button. And what we're looking for is we're asking the millions of virtual electrodes, show me only those, so we're sensitized, show me only those voxels that show decreases in beta band power over the half a second before you press the button. So we ask people to press a button whenever they fancy it, and we interrogate the window just before they press the button, which is when their sensory motor cortex is desynchronized and ready to execute the task. And that's how we do motor mapping. So you can see that based on the time domain, we had something like a spike detector. So that might be called something like a map of dysfunctional brain. Based on the spectral domain, the frequency domain activity, we're able to map eloquent cortex, in this case the motor strip, and then just for a bonus, we can put them all together with MRI as part of a complete pre-surgical mapping application. So it's essentially combining structure, which we had from MRI, with both function and dysfunction, one coming from the time domain and one really coming from the spectral domain. And again, if we want to think about this, I was able to interrogate a different window of time for the motor activity, the 500 milliseconds before the button press. But we could also, in the word experiment, look between 200 and 700 milliseconds after the stimulus. So you can imagine that you can look at different moments in time and say, which are the areas of the brain which are most active at different moments in time during a task. So here's a task where you're presented a word on a screen. And if you only say which areas of the brain are showing event-related desynchronization over the first third of a second, it'll be mostly visual cortex because you're mostly seeing stuff. And if you say, aha, but after that, after the first third of a second, what happens between, say, about 300 and 600 milliseconds? Do the same experiment, and what we see in the left hemisphere is posterior temporal, inferior frontal, language-related activity. And then supposing we had created a task where you read the word, you think about the object, and then you perhaps raise your hand to make a response. And we ask later on, just around about the time of making the response, so that little window of time, 600 to 900 milliseconds, which areas of the brain are most involved then? It turns out that it's the motor strip. So what we have here is essentially time lapse, or something like frames from a functional brain movie. So we're no longer thinking about functional brain activity as a static thing. It's a dynamic thing. It moves from visual cortex to language areas to motor areas as the demands of the task vary. So I'm going to talk about a little bit more in the way of applications. We've already had a nod to the time domain kurtosis showing up as useful for epilepsy. Low frequency power is showing up as useful in trauma. I'm going to talk a little bit about some timing things and some frequency things, focusing primarily on autism, but not exclusively on autism. And you can think of that as an entry point into neuropsychiatric disorders in general, where structural findings have been lacking. So here again is a movie of the spatial and temporal pattern of activity associated with hearing a simple auditory stimulus. And you saw that first there was nothing going on, and then there was a surge of activity after about 50 milliseconds in the superior temporal lobe, another surge around about 100 milliseconds. So we're able to characterize the time course of this activity. And what if we turn that into something more like an ERP, we're able to go into the brain, into the auditory cortex, and say, show me the time course of auditory response when presenting a simple stimulus. And what we have found over and over again is that there is indeed, of course, in this pink trace here, an evoked response component that's characteristic of a typical hearing of a sound. That's evident also in our psychiatric populations and developmentally disordered populations, including autism. But in this particular case, whilst the morphology is very similar, the exact timing is shifted in this case by 10 milliseconds or so. So there is a question which is, is this 10 millisecond delay something that's really biological in basis, or is it just an epiphenomenon? It's useful. It certainly shows up in autism. It shows up really independent of the type of stimulus that you use. But it's not really it's not bad, but it's not a very good diagnostic tool. But nonetheless, it still begins to get the claim of this word biomarker because it's some sort of measure, organic measure, that can distinguish autism from some other disorder. So that makes us think a little bit about this question: when does a marker become a biomarker? Because it's not really biologically based at this point. It's just a timing thing. How could it happen? And in this specific case, what is the biological basis of this delay? So that's when we have to step aside from electrophysiology for a moment, invoke one of our other technologies, and this, of course, is MRI, and particularly diffusion MRI. And diffusion MRI is, as many of you are familiar, it's the technique that has gained widespread acceptance for the imaging of celery, primarily for imaging of the fibrousness of celery. So if you want to know how tough your celery is going to be and chewy and get stuck in your teeth, the easiest way of doing that, you'd think, is to eat it. But actually, the best way to do it is to put it in the MRI. Now, of course, if you can image celery fibers, you can also image white matter fibers. And so if you think about this particular technique that's so good for imaging celery, it's also very good for imaging white matter, which allows us to interrogate the white matter of the auditory pathway. Now, that's important because if you somehow were to have a problem along your auditory pathway, you might expect that your auditory cortex response out here in the temporal lobe, that cortical response might be delayed if something went wrong between the ear and the brainstem and the thalamus and the cortex. So in studies of that, we were able to show that there is, in fact, a development of the fibrousness. It becomes, if you like, more fibrous with age, typically. And that as the fibers become more fibrous, as these imaging properties change, suggesting a normal pattern of maturation, what we see is that the cortical response latency, so this is the timing from the electrophysiology, becomes faster, or let's say more mature. And that actually turns out to be true pairwise. That is to say that the quality of the white matter predicts the timing of the cortical response. In this case, the x-axis is something from the MRI that relates to the fibrousness. So this is very fibrous. This is immature. This is very late response. This is a more mature early response. So as you'd predict, as the white matter gets better, the responses get faster. And what that led us to was some sort of conduction velocity hypothesis that the white matter is maturing, maybe that the myelin is compacting, whatever it is, to more effectively communicate its signals, resulting in a shorter latency. We weren't the only ones to observe this. Steve Stufflebeam in Boston was able to do this in a population, showing, again, an association between diffusion imaging or fibrousness imaging properties in different structures and visual evoked response latencies. And this also turns out to be an international finding. Colleen Dockstede at Sick Kids in Toronto has found a similar thing. So there is some sort of association here between microstructure, quality of the white matter, and timing of evoked responses. Well, so in my particular study, it was really maturing because we looked at 6-year-olds, 7-year-olds, 8-year-olds, 9-year-olds, 10-year-olds. In these other two studies, the reason for the differences in quality are less clear to me, what their population differences came about from. But we used development. Yeah, they use the same measurement. So I mean, you're right to interpret it as quality, but we modulated the quality by developmental maturation. So I think it was the sort of a natural lab type experiment. David, I think you had a question.
I had a question about the amount of time here. So you're measuring the fractional anisotropy in the white matter, and you're seeing these time differences that correlate with that are like 40 milliseconds. Is it reasonable that the conduction velocity of an unmyelinated or immaturely myelinated fiber would be that much slower?
Well, so of course you're stealing the punchline a little bit.
Sorry.
That's okay. So usually I would describe this in terms of driving from Philadelphia to New York. So why don't we do that? So as you all know, especially those of you who live in New York who ever go to Philadelphia, the biggest problem getting from Philadelphia to New York is I-95. And I-95 is like the white matter here. And so, you know, if I set off for Philadelphia to a meeting at the Simons Foundation in New York, I will be late to that meeting, and I will blame it on traffic on I-95 or construction on I-95 or something wrong with the white matter quality of I-95. That will get me late to my meeting. However, there are certain occasions on a Sunday afternoon when I-95 is behaving quite swimmingly and you're driving along very, very fast and you think you're going to make your meeting on time. White matter's okay. You're still late to your meeting. And you're still late to your meeting because of something else. And the thing that you're still late to your meeting because of is the Lincoln Tunnel. And the Lincoln Tunnel, in this very rapidly getting out of hand analogy, is the Lincoln Tunnel is synaptic transmission. So you can motor down the white matter. Conduction velocity accounts for some of the delays. But synaptic transmission counts for other amounts. And what's really interesting is in different people, you have different proportions of the conduction velocity explanation and the synaptic transmission explanation. And it might well be the case that you were able to, and this is now speculation, that we could tease apart some of the heterogeneity of our findings based on whether you've got more of the white matter type problem or more of the synaptic transmission type problem. But you can certainly have a few milliseconds based on conduction velocity. So you might end up with something like that. Oh, and this is then the last thing, which is, again, since we're at the Simons Foundation, to show this result, to prove that, or not to prove that, to further support the idea that there's a biological basis to this timing delay that we see. If you happen to be missing a chunk of chromosome 16, one of the striking findings from this cohort is that your M100 will be delayed, as though there's something in the genetic material that area of 16p contains that is necessary to make a perfectly fast, normal M100 response. And if you don't have that because that particular region has been deleted, you'll end up with a delayed M100. So that's towards biological basis. So remember before I said there were lots of reasons to go into the brain. The first one was clinical, anatomic interpretation. But the other two, signal-to-noise increase and projecting away those pesky EEG susceptible artifacts, well, it turns out that those two are something to do with the mathematical combination of the signals, the spatial filtering, and those advantages persist even when you don't have an individual's MRI, and you, in fact, just say, I want to project into some space that's a bit like my individual. Now, this is a bit counterintuitive. We're now talking about not having an MRI to do our source localization. So what do we lose? We lose our anatomic interpretation when we use some sort of average or representative MRI. That's not good. You certainly wouldn't want to use an average brain before planning surgery, so you sacrifice that. But you do retain the signal-to-noise advantage, and you do retain the ability to project away different artifacts. That means that we can keep most of the advantages of source-modeled MEG, even in populations in whom it's impossible to get an MRI. And those situations might well be particularly low-functioning individuals, particularly young individuals who really don't like going into MRIs. And those communities are, in fact, poorly studied in our imaging field because they're difficult to get in the magnet. And because they're difficult to get in the magnet, we don't study them, so we don't know anything about them. And so I think that this is a feature that we really haven't talked about very much in the literature, which is that the MEG signals, and to a certain extent the EEG, but really the MEG signals can go through all of this signal processing pipeline and yield the high quality, high signal to noise, artifact-free signals that we want, even in populations where we couldn't get them into the magnet, allowing us to study the broader spectrum of disorders like autism, including also developmental delays, children with low IQs, and very young children. Proof of that, not proof, but supporting evidence of that comes from a slide given to me by Chris in the audience, so I hope I don't get this wrong. This is a 6-year-old child who actually does have an MRI. So this is a relatively compliant 6-year-old. And we do our usual stimulus. We deliver an auditory stimulus, and we get a nice cortical response in the left and right hemisphere. After we go into the individual's brain. So here we are in the superior temporal gyrus of that individual. Then we say, well, let's just suppose this wasn't a 6-year-old child who's compliant, but this was like most of the other 6-year-olds who's not so compliant, doesn't want to go in the magnet. So let's throw away their MRI and pretend we didn't have it. And if we didn't have their MRI, we just have to use one that we got from, downloaded from the web, somebody else's MRI, or an average MRI or template. And when you co-register to a template MRI and you still go into brain space, but now not that person's brain, just a brain, we can still get these nice high signal to noise, source-modeled auditory evoked responses with exactly the same peak latencies, 122, 138 as it happens to be. So the point here is that we're able to leverage many, many, many of the advantages of going into source space, even in a non-compliant or low-functioning population. Why that's interesting is, of course, we'd like to start seeing whether we can get these evoked responses in very young children, where we would probably never be able to get a good MRI, and to see whether this signature is in fact a diagnostic signature for autism. Now, clinical diagnosis is made at 2 or 3 or 4 years old, so we need to be able to make that in a younger child for it to be a useful diagnostic biomarker. So consequently, we need to be able to leverage the advantages of signal processing, that is essentially the signal averaging of the multiple sensors and the rejection of artifacts, but in the younger child without an MRI, and we believe that that's possible. Interestingly, as I said before, R², of course, is our enemy here, so we had to build dedicated MEG hardware to do this as well, wherein the MEG detectors are much closer to the brain of the small child. But this wouldn't be enough if you couldn't also take advantage of all of that signal processing that we just heard about. Okay, one more timing thing to talk about, and the story is very similar here, so we'll go over it relatively quickly. The idea here is, again, that the timing of an evoked response relates somehow to the structure of the brain and can be clinically useful to us. So here's what happens when you hear the syllable ah. And if you hear it again, you get the same response. You hear it again, you get the same response. Hear something different, you get a hearing response and also some sort of, oh, that's something different, a change detection response. And simply put, you can determine how much of the brain activity is sensitive to the change itself by something like subtraction. And you subtract to get these things called difference waves, and the difference waves now have peaks in them. And this is not the peak of hearing, this is the peak of detecting a change. So this is called the mismatch negativity or magnetic mismatch field. And the point here is that what we're able to see here is the time at which you registered, or your brain registered, that the syllable had changed. Now this is pretty important because I'm busy talking to you with lots of syllables, and they're changing every quarter of a second or so. So I'm speaking at about 4 hertz. So that means you've got 250 milliseconds to process the first syllable before the next syllable comes along, and the next one, and the next one. So if you're a little bit slow processing syllables in a constant stream of auditory speech, it's going to be really difficult to keep up. And that's exactly what we see on the autism spectrum. yellow and red are autistic children compared with green, typically developing children. And what we see is that there's a delay in this characteristic neural response of change detection to a stream of syllables.
This is an N of 2.
Oh, no, there's an N of about 100 in this paper. What I'm showing here is these are the error bars on the latency of the mismatch field. This is autism. This is typical development. Beyond that, I've stratified. I'm getting there. Because that's a very important point. So you know that the autism spectrum is very heterogeneous. So we've divided up this population of children with autism into those who have clinically significant language impairment, according to one of the neuropsych scales for language, and those that don't. And the yellow ones certainly are on the autism spectrum, but don't have terribly bad language problems. So we use the core language index of the self-4, for those of you who are familiar with that. So that's a bit like an IQ test for language. And if you have a score of between 85 and 115, that's about normal, kind of like about normal IQ. And that's what these children had. Some of the children had clinically significant language impairment, meaning their score was less than 85. That's the children in red. So these have a language impairment. These don't have a language impairment. And what you notice, of course, is that the severity of the delay is worse in those that exhibit the more severe clinical phenotype. That actually is true on a scatter plot. Latency, timing of the change detection response on the y-axis, clinical score on the x-axis. 100 is the median. So our green, our typically developing children, are mostly around about somewhere between 90 and 110 on this scale. Yellow are children on autism spectrum who are not significantly language impaired. You can see that because they have scores that are essentially over 85. And the red are the children that have the language impairment. The point here is that this neural measure appears to be prognostic, or at least appears to relate to the severity of some domain of phenotype, in this case, language ability. Yeah.
Could you go back a slide?
I don't know. Yes.
Yeah. So I'm looking at this and I'm seeing those three curves, the time courses at the top, kind of going up together. If anything, the red one starts earlier, and where the green one has its peak, all three of them. It's hard for me to see the yellow, but it looks to me like all three of them are right on top of each other, and then the yellow one and the red one keep going. So I'm wondering how you think about that. So if the idea is that there's a signal that indicates the change, it seems like it's there in all three of them, and then it's bigger in the yellow group and the red group, but it takes longer to get to those yet even bigger values.
Well, I think what you're looking at here, we should remember that these are grand averages. So this is not these are not from representative individuals. This is the grand average of the population. So you have some amplitude-related sort of dispersion effects or artifact from that. I think it's kind of a red herring. If I were to show you a group of, when we use this on an individual basis, we certainly don't see that amplitude trend showing up, and you do see the latency as really quite a powerful way of classifying the groups. So I think that that's probably just something to do with grand averaging and not a meaningful. If you go to the paper, there really are no meaningful amplitude effects.
So then I guess that's what I'm asking. So when you look at data like these, you look at the timing information
Yes.
as being reliable, and the amplitude values you don't really trust.
That's entirely correct, because it's if you do a correlation of, for example, center level and source modeled data, amplitudes they vary with a whole bunch of things, like how good is your model, how good is your signal to noise, how far are you away from the center. Latency is quite hard to mess up. That seems to be an intrinsic feature. The timing is the timing. You may lose some precision in your sensitivity to it, but it seems really robust. And as you know, one of the reasons we harp on about latency all the time is it's there to be measured. And it really is robust to measurement technique, whereas I think there are a number of factors that you can imagine modulating amplitude that really don't get to latency. Yep.
So in the same last slide, what's actually happening afterwards? So if you look at the dip afterwards, there's a big difference between the three waveforms. So I'd be more interested in knowing what's going on there. Or what do you think is going on there?
Yeah, so repeating the question,
Speak into the mic. Push your microphone button, and then put your mouth near it.
but let me repeat the question. I think what's being asked is, why do I stop being interested at the first peak in this difference wave when there's all sorts of interesting things going on later and later and later? I think that's absolutely true, and that's actually true of electrophysiology in general. The first 50 milliseconds or so defines the first decade of my career. The next 50 milliseconds or so, the next decade. The problem is these are subtraction waves, so you get into wrinkles associated with that. Later stuff is fickle. It's very highly variable across individuals. And it's much more interesting, because the later stuff is probably higher order cognitive processing of what to make of that difference that I've just spotted. I wonder what it means for him to have said ooh. Is that a word? Is that something interesting? And that's the sort of stuff that's going on here. And if I were to live to be like 200 or 300 years old, we'd probably get all the way to the end of this. But hopefully somebody else will pick that up. So yeah, generally speaking, early stuff is better contained, and later processing becomes more and more variable. Of course, that's what we're studying, but I don't have any interpretation right now. So of course, here's something that's interesting. This is the Arcuate fasciculus, again on the celery imaging. So this is the white matter fiber on the left hemisphere that is associated with language functions. And interestingly, looking at this quality factor, or in development maturation, but here probably quality, we notice that the populations typically developing autism spectrum without language impairment, autism spectrum with language impairment, show the same stepwise pattern, where in this case low is good. It's the same as short was good before. So what we're seeing is something that's strangely analogous between the quality of the white matter and the timing of the evoked response. That's not bad, because we've just seen that, and we've made a conduction velocity argument for something sensory. Now we're talking about something a bit more cognitive, but we have a similar pattern of association between white matter dysfunction and timing of in this case, the change detection response. It gets even better when you look at this, which looks strikingly familiar, because you've just seen one where the y-axis was timing and the x-axis was clinical score. Now the x-axis continues to be clinical score, but the y-axis is quality of the white matter of this language pathway. So you're struck by the fact that the red are up here on the left, then the yellow, then the green. The point being that in this case, the quality of the white matter also seems to be associated with the language function. And to a certain extent, that's pairwise true. Again, saying that the quality of the white matter accounts for at least some of the variance in the latency of the evoked response. So back to David's point, it doesn't account for all of the variance, because there's probably more to generating a mismatched field than just the diffusion properties of the Arcuate fasciculus, but it accounts for some of it. So to stop the discussion on timing, just a simple thought. To resolve intrinsic timing differences that are of the order of milliseconds, you need temporal resolution of the order of milliseconds. So this is, given that we think these differences are biological in origin and clinically interesting, we need to have electrophysiological probes. So again, it's kind of preaching to the choir, but this is not the domain that we should be studying with other modalities like PET or fMRI. If you really need to find things that are happening on a millisecond scale, you better measure things with a millisecond scale. Now, switching gears a little bit now to the other thing that's afforded by high temporal resolution is some spectral information. So instead of having an auditory or stimulus evoked response like this, we can decompose it into its, could be Fourier components, in this case could be band limited energy, or we can make these time frequency, slightly like spectrogram plots, where we have the x-axis as time, so nothing's going on, and then at 0 a stimulus comes on. Red means a lot of power, so there's a lot of low frequency power straight after the stimulus. But there's also some high frequency, there's also some high frequency power around about this 40 hertz so-called gamma band. And notwithstanding the previous discussion about there should be an elevated baseline or a broadband signal in a number of stimulus paradigms, I think it's very clear that whatever's going on around 40 hertz is significantly up above whatever may be happening at other frequencies. So yes, there isn't a unique interpretation to this 40 hertz elevation, but it is specific to that frequency band in addition to whatever else is going on. So we're able to take our signals and decompose them spectro-temporally. Why would we want to do that? Well, we've already seen one example that just looking at low frequency power is in fact diagnostic for us. It shows when we can localize it and say that's where the person hit their head. That's traumatic brain injury. Or, as I just alluded, we can evoke these rhythms by some sort of stimulus, or frankly by some sort of cognitive challenge. And then to take it very simply, if you're believing that these are oscillations, there are only three things that can go wrong with a wave: phase, amplitude, and frequency. There isn't anything else. So either, if there's a pathology of your waves or of your oscillation, the pathology is likely to show up in terms of a phase problem, an amplitude problem, or a frequency problem. And sometimes pathologies of oscillations are being called oscillopathies. I just feel like I should say that. It's not a Scrabble word. It's got too many L's and whatever. It's too long, but we'll just say oscillopathy a few times. So here's a study that we did. It's a multi-center with the group in Cardiff, where we were able to show that if you deliver a visual stimulus, and that's the visual stimulus, it's a grating, just stare at it. While you're staring at it, your visual cortex will oscillate. Well, not the cortex won't oscillate, but you will have cortical oscillations in the visual cortex at the gamma band. You'll have a burst at the beginning when you first see it. That's called the transient response. And then you'll have a sustained oscillatory response as long as that stimulus is on. When it goes off, the response will stop. Interesting feature of this gamma band activity is it has a sort of central frequency. It is a bit broadband. There's a bunch of red, but there's bright yellow in the middle. And the y-axis here is frequency. The x-axis is time. So this is characterized by a kind of a frequency that particular individual's brain is really reverberating at as it's processing this grating stimulus.
Those two separate panels.
This is a single panel that for some reason has a white line going across the middle of it. No really good reason. It's kind of, if the white line had been a tiny bit higher, it would have been to point out the frequency of the gamma band oscillation. So everybody, all of you doing this at the moment are having gamma band oscillations in your visual cortices. What this study was about was to say, are they all the same? And it turns out they're not all exactly the same. Exactly the y height of that yellow blob depends on things. And the thing it depends on most, the thing that the frequency depends on most, is your age. So when you're young, that's up at 60, 70 hertz. And unfortunately, when you're my age, it's down here. We're losing about 1 hertz per year. So if you thought you really can't afford to be listening to this talk, you really can't. You're losing, you're losing, your brain is losing the ability to drive fast oscillations at approximately 1 hertz per year. So an hour is going to be fine. But so this is basically saying that whatever machinery, whatever local circuitry you need to generate these oscillations becomes progressively less effective. And this is when I was saying what goes wrong with an oscillation. Well, one of the things that goes wrong with it is the frequency of it, the rate of it. You simply can't oscillate as fast as you get older. That's just, yeah, there you go. So we've talked a little bit about this before, but here's just to reiterate, if you have a stimulus-locked oscillation, a little burst of evoked gamma power. Every time you do it, it'll happen at the same time. And so when you average that together, you'll get a nice ERP, and you'll have a lot of phase stability. That is to say, that a lot of synchrony. If you've got a nice, tight system that's ready to respond to the challenge of a stimulus and deliver its burst of gamma, and it does it in a very reactive way every single time, you'll get a high similarity, which we define as phase locking, high phase synchrony, sometimes called intertrial coherence. If, on the other hand, your machinery is a little bit more kind of sloppy, and you get a stimulus and you have a burst of gamma, you get another stimulus, you have a bit of a burst of gamma, and you don't do that in a very tight and reproducible way, that's will manifest as a loss of trial-to-trial phase synchrony, a dropping of the phase locking factor, intertrial coherence, or whatever we mathematically describe it as. The point being, this is a sensitive indicator of the quality of the local circuitry to do its thing and respond with a burst of gamma. So it doesn't have to be that the frequency will drop off. It could be that the quality of the response just isn't there, trial to trial. The reproducibility isn't there. That's going to be important because what we see, again, in autism is whilst when you have an auditory stimulus characterized by a big low-frequency evoked response that we've just measured the latency of, there's something else that you get, which is a nice, tight burst of gamma oscillation. It's like, woohoo, I just got a stimulus. And every time you deliver a stimulus, you get the same burst of gamma. And this is a plot now of time, frequency, and this is the gamma band, phase quality, something that's evident to us from the signals. And what we notice on the autism spectrum is that there's a conspicuous decrease in the phase synchrony, or in this case, the phase locking, we call it, but the quality of that gamma band oscillation. It may just not be there at all, or it may be that it's not able to be phase locked in such a tight way. And so we think this is a sensitive indicator of the local circuitry being not quite right. And it turns out that that correlates with, in this case, with language function as well. The quality of that oscillation, the ability to generate a good gamma oscillation, has a clinical correlate. So that makes you think, ha-ha, a clinical correlate. That sounds something like you'd want to have. Does having a clinical correlate make you a biomarker? And I'd say no. At this point, it's still a marker that's of clinical utility. It's not really a biomarker. What's the biological basis of it? So perhaps the biological basis lies in something that is going on inside that local circuitry. That is the processes of excitation and inhibition, perhaps between inhibitory interneurons and pyramidal cells, perhaps mediated by that excitatory neurotransmitter glutamate and the inhibitory neurotransmitter GABA. But we can't measure GABA and glutamate with electrophysiology. That measures evoked responses and timing and spectrum and stuff. But we can measure it back in our MRI. We don't do the celery sequence anymore, but we do some chemistry. And this chemistry is called spectroscopy. There's a particular flavor of spectroscopy. We can talk about the details of that. But the bottom line is that with this fancy spectroscopy, we can take advantage of the chemical nature of GABA. That's a GABA molecule in chemical formulae. We can look in auditory cortex and say, show me your GABA. We can measure the amount of GABA in the auditory cortex or in the motor cortex or visual cortex, wherever we want, and see if we can get some estimation of an imbalance in the neurochemistry that might be supportive of what we're seeing in terms of oscillopathy. So this was first done by the group, again in Cardiff, Suresh Muthukumaraswamy, who did this in visual cortex and showed that the predictor of the quality of the visual gamma band oscillation, other than age, another predictor of the quality above and beyond age was the level of GABA, the neurotransmitter in visual cortex. So he's able to show here that essentially the GABA levels will determine the frequency of the gamma oscillation. So this is saying that there's a neurochemical basis for the oscillation. Yes. Can I show you in like 2 slides? We then thought, that's really interesting. We'd like to replicate that study, but you can't replicate studies. Anyone who's tried it knows you can't get them published. So we replicated it in another system, which is to look in the motor system. And this is our time frequency associated with pressing a button. So the button gets pressed, and there's a burst of gamma as well as the other oscillations that we've talked about. And it turns out that the biggest predictor of that gamma burst is, in fact, the level of GABA in motor cortex. So we put our region not in the visual cortex that Suresh did, but in the motor cortex. And we're able to show a coupling between neurochemistry and neurophysiology in the motor cortex as well, leading us to remember that Rubinstein and Merzenich at UCSF had suggested that autism might in fact be, might have a neurobiological basis in an imbalance of excitation and inhibition, which was an idea they primarily got from looking at the comorbidity with epilepsy and knowing that epilepsy is associated with an imbalance in GABA and glutamate. So perhaps autism is as well. And that allowed us to think about, well, perhaps we can probe in typical development the intactness of this circuitry with intact levels of GABA, leading to good oscillations, which, by the way, might later on resolve as nice coherence and connectivity. Whereas if something has gone wrong with this picture, it might show up as a disturbance of neurochemistry, which might show up as a poor quality oscillation, and we've already seen that we have those, which might in fact show up as poor region-to-region connectivity. So is there any support for Rubinstein and Merzenich? Well, here we did the experiment where we measured GABA in auditory cortex in a bunch of individuals. And to answer your question a little bit, black lines are typically developing controls. Gray lines are children on the autism spectrum. We're looking at relative GABA levels normalized to local creatine. And what we notice is that the contrast between typicals and autism, we see no difference between them in visual cortex. We see a small difference in motor cortex and a larger difference in auditory cortex. But to answer your question, absolute levels, there's not enough difference there to really know. There are regional differences. People have done those studies with more quantitative methods. But the point here is that not only is there evidence for GABA imbalance or GABA-glutamate imbalance in autism, but it's different in different parts of the brain. And that's very consistent with the idea of different types of phenotype, that we've got regional variation in a similar mechanism, that is EI imbalance leading to poor oscillations. But because there's regional variation in it, that could lead to, because of functional specialization, could lead to regional differences in the impact of that on phenotype. And this has now been shown by 2 or 3 other groups who've also shown decreased GABA in the kind of in the frontal lobe, not in the deep brain. Don Rojas has also shown it in auditory or superior temporal areas. So I wouldn't say it's a highly replicated finding, but it is support for the notion that there is atypical biology or neurochemistry in these children. Just grouping it together, the idea here is that if you have low GABA, you'll have low gamma power, and that's characteristic of the autism spectrum, higher GABA levels associated with better quality gamma oscillations in typical development. I just want to spend a couple of minutes on another reason why we pursue biomarkers. Another reason we pursue biomarkers is we are increasingly being made aware of risk genes and genetic factors for disorders and disease. So there's a lot of genetics going on that leads people to make mice that have some gene knocked out or different. But the question is, how do you evaluate whether that mouse is really helpful to your understanding of the disease? You know it doesn't have that particular gene, but how can you characterize it? You can characterize mice by their behavior, and that's certainly being done. But it's relatively difficult to mimic human behavior in a mouse, and perhaps not even the right and most important thing to do. What we could do is perhaps say, does this mouse model with a particular gene knocked out, does it recapitulate something that we observe clinically? Not behavior, that's too difficult. The stretch between one gene and behavior is probably too big a bridge to cross. But how about it recapitulates something in the brain of the mouse that we saw in the brain of children on the autism spectrum or with the other developmental disorders? And so we think there's a real role for these types of signatures, types of measurable neurophysiological things like latency and kurtosis and complexity, as a bridge between the clinical environment where we observe those things in children with the disorders and these candidate mouse models. And if a mouse model recapitulates that brain-level phenotype, then we should at least feel encouraged that that is a useful way of gaining some understanding in the preclinical environment about that aspect of the phenotype. It doesn't mean it's a mouse model of autism. It means it's a mouse model of delayed M100s, and maybe we can learn something about delayed M100s. Case example of that is from Steve Siegel. This is the valproate mouse. It's a model of, has been used interpreted in the context of both autism and schizophrenia. It has a bunch of atypical behaviors, but what we find interesting is that it also has a delayed auditory evoked response. Very parallel to the top there is the human delayed auditory evoked response, and that is the mouse delayed auditory evoked response compared with the saline control mouse. So this really suggests that the timing parameters are an electrophysiological trait that allows us to bridge the clinical and preclinical environments. It's not just the timing, but also the quality of the gamma oscillations, the phase of the gamma oscillations. Again, at the top, human data, typically developing humans in black, children on spectrum in gray, showing a decrease in the quality of those gamma oscillations, the phase synchrony. And what do we see? Saline mouse in black, valproate mouse in gray, a decrease in the quality of the gamma oscillations. So that perhaps is a model that could be investigated further, and these are traits that could be used to, for example, monitor experimental therapies and to screen them through more effectively before they get into clinical use. So we're very comfortable that this suggests a set of traits that can be translated between the two labs. And here's a reason why you might want to do that. This is just a one-off example, but one view of the utility of a biomarker is actually measuring response to a therapy. And you'd like your biomarker to be tuned to the mode of action of the therapy, because otherwise, it's just an epiphenomenon. And so we have here gamma phase synchrony in a child with autism at baseline. None. There's no gamma phase synchrony there. And after 12 weeks of a synaptically targeted drug, lo and behold, the gamma phase synchrony re-emerges, suggesting again that this is the type of way that you could use these measures to monitor the activity of, in this case, a GABA-acting drug. Now, of course, if you were to give aspirin or something like that, this would be a very silly biomarker because it has nothing to do with the mode of action of the drug. So it's important to match your biomarker to the mode of action of the therapeutic. Hello. Well, it's a huge question. I think that one has to take the position that pathology is bad and that disease is bad, and that if you have a marker that's associated with the disease and poor clinical behaviors, that's bad, and that if fixing, and if that marker points you towards a putative biological mechanism for the underlying the bad behaviors, like poor synaptic transmission, poor local circuitry, that's bad. And if it then suggests that that's arising from an imbalance of neurochemistry, then we could say that imbalanced neurochemistry is bad. And that if you then restore the balance of the neurochemistry, bringing with it the biomarker and ultimately the behavior, that's good. So you don't know per se that fixing a marker, or it's a very similar situation in epilepsy. You don't fix the electrophysiology per se, but that's why I make such a big fuss about there being biomarkers, not just markers, because they're pointing us to a mechanism, or to at least to a substrate that that has biological plausibility to it. So ultimately, the proof is in the pudding. You don't know that it was worth doing it until you fix the behavior, but you have no idea how to fix the behavior until you have a biological target. So I think there's value in identifying candidate targets to be explored, perhaps in the preclinical setting, as, hey, what happens if we pursue that biological target versus another one? Do we recover behavior? So per se, I'm not going to make a sort of judgment about a biomarker being good or bad, or is it good to have a gamma oscillation? All that we know is that in typically developing people, there are these gamma oscillations, and that they are deficient, and they can be recovered. So yeah, I don't want to suggest for a moment that it's good to have a big evoked response versus a small evoked response. There could be all sorts of other things going on. But I think that what's nice is to retain a biological interpretation for it, to get a biological target, and keep an eye on the prize, which is ultimately behavior. So if we wanted to get some take-home points from this so far, I would say that electrophysiology in general reveals these millisecond-scale anomalies in a variety of disorders, which are often invisible to other modalities, and that we have seen a variety of examples that capitalize on the temporal and the spectral or spectro-temporal features. I think electrophysiology is great, but I have struggles, as we just got to, really making the interpretation of, let's say, a quality, a value judgment about the electrophysiology without help from some biology. And that's usually where we get converging evidence from MRI or MRS to help us with the biological interpretation of the electrophysiology, because you're quite right, without that, it would just be a thing that happens. Now it's a thing that happens because of bad white matter, or a thing that happens because of ineffective synaptic transmission. Now I can begin to put some value judgment on that. And make the point here, of course, that these markers are not just about diagnosis, but also about measuring response. So I've got 20 minutes left, so I want to kind of bring it all together through the study of connectivity. And connectivity means doing all of those things that we've just done, measuring the signals in different parts of the brain, but comparing different parts of the brain with each other. And the idea is essentially that if two different parts of the brain are doing something similar, then maybe they're doing it somehow together, like different instruments in an orchestra, and that somehow that is that's evidence for network scale function as opposed to discrete local functions. So measures of connectivity could be as simple as coherence, just like is something going on in region A the same as something going on in region B, and do something like a correlation coefficient. This is work from Chris again, showing oscillations, alpha band oscillations, so narrowband filtered oscillations, in blue in the calcarine cortex in the occipital lobe, in red in the thalamus, connected, as it happens, by white matter. And this is during some sort of simple alpha challenge, eyes open, eyes closed. And what you'll notice is that there is alpha power both in the cortex, in the calcarine cortex and in the thalamus, and that it comes and goes with a similar time course, and that there's a phase lag. So these oscillations come and go, broadly speaking, together, but the difference here is that there's a 13 millisecond phase lag between the thalamic source and the cortical and the superficial cortical source, suggesting that these two oscillations are coupled to each other, but they're coupled by some sort of propagation mechanism which introduces a delay, and let's call that white matter. So if you wanted to measure the conduction velocity, I suppose you take that distance and 13 milliseconds and divide one by the other. So that's essentially a simple measure of correlation between two areas of the brain, suggesting, and because the thalamus comes first, it suggests that perhaps the thalamus is driving the cortex in having these alpha rhythms. We know that's true, but this is supportive evidence for that. So that would be like a simple coherence or correlation method. Or it might be a bit more complicated than that. It may be that the two areas of the brain that are communicating with each other are not just having a nice sinusoidal alpha rhythm, but are doing completely different things. But that every time I raise my left hand, somebody over there scratches their ear. That's not something that will be revealed by correlation, but it is nonetheless the case that we are somehow synchronized. So there's a whole family of synchronization, generalized synchronization methods, and one that we use is synchronization likelihood, which essentially says in two different brain regions, every time something happens in one of them, does the same something. Oh, sorry, let's say this again. If there's a type of an oscillation, an event, a deflection, a thing that we see in one channel, at various other times in these other boxes, we see something very similar. So that is to say that there's one part of the brain does something, and then a little while later it does it again, a little while later it does it again. That's just what it does. In another part of the brain, how often does whatever that part of the brain does recur at exactly the same time as it did in the first region? And that's to say they don't have to be the same activities, but it's basically saying, are they often happening at the same time, whatever it is that they're doing? And that's essentially synchronization likelihood. And that's a way of more generally allowing different parts of the brain to be computationally connected without demanding that they are correlated with each other. They are temporally coincident repetitive events. And then you can make connection maps based on that. And just for fun, instead of making connection maps, we make connection movies, because we can do that analysis every few seconds. And this is a pattern of the connection strengths between different parts of the brain every 5 seconds. So this is essentially imaging transient connections in the brain. It's as close as we've come to imaging fleeting thoughts. That is correct, although but we have actually bins of time. So there's a tolerance to a time offset is still included in this, but it's not explicitly tested. So what you're just seeing here is either a lot of noise or transient connections, because we're able to make these connection descriptions every 5 seconds or so. Analyzing that, of course, is difficult, but one of the things that Yasser did in our lab was to look at those sorts of connection strengths and compare that with a temporal quality of the signal, not the latency of an evoked response, because he was interested in what the ambient brain is doing. So there's no evoked response to have a latency. Not the kurtosis, because these are not children with epilepsy. They don't have transient spikes. But he still thought that the signals from different parts of the brain would be characterized by different complexity. Complexity is something like predictability. So essentially, complexity is the opposite of predictability, so something like entropy. So he measured multiscale entropy, and he also measured the connectivity using this synchronization likelihood. And we did it in children with typical development and children with autism. The reason for using this is that they very kindly used these slides on the advertisement for this seminar. So I thought I would explain what they were, because they're a little unusual. What we're looking at here is connections essentially between different parts of the brain. Connections which are significantly different in the population with children with autism. So significantly stronger in autism would be red, significantly weaker in autism is blue. And again, we've done this in band-separate ways, so the richness of the data here is immense, but this is low frequency, delta, theta, alpha, beta, gamma, and broadband signals. And we're looking at those types of connection which are stronger in autism, tend to be only very short-range ones in very low frequency band in the frontal lobe, at the delta band at the top in red. And also some parieto-occipital connectivity seems to be stronger in autism, this functional connectivity, whereas almost everything else, including a lot of long-range connections, even in the broadband, seem to be weaker in autism or stronger in typical development. Wow. So that's basically saying we've got evidence for frequency-dependent, length-dependent differences in functional connectivity in autism. But how does that relate to complexity? So this is the idea for complexity. It's basically how predictable is the signal. So for example, in this particular waveform schematic, you have a green dot, then a pink dot, then a red dot. And then you have a bit more waveform. How often do you see green, pink, red in the pattern? Well, you see green, pink once there, and green, pink once there. But green, pink, red only occurs less frequently. So this is basically trying to describe the chaos or the entropy of a signal. How predictable is it, or in this case, how unpredictable is it? And so one can formulate complexity a variety of ways, but we like to use this entropy measure. So this is something that's, again, extractable from the MEG. It's a time domain quantity where you can say, if this was a smooth sine wave, it would have very high predictability, very low complexity. If this is a crazy sort of signal, it's going to have a lot of complexity because you won't know from one point what the next one is going to be. The funny thing is, when you look at group differences in complexity between autism and controls, what we notice is that in the red or yellow areas of the brain, complexity is higher in autism. In the blue areas, complexity is lower. That is, the signals are simpler in autism. And that's on the right. What's funny is that on the left, these are not complexity maps. These are the connectivity maps, just smoothed to be the same color scale. And what we're seeing is, this is exactly what I showed you before with red frontal hyperconnectivity in autism. These are almost exact complements of each other. That is to say that whether it's the delta band, the theta band, the alpha band, or the other bands that I don't have enough space for, where you have high connectivity, the red at the top, you have a low complexity. And where you have low connectivity, you have high complexity. And they correlate really very nicely with each other. This is basically the difference between typicals and autistics, are where in those regions where connectivity is greater in autism, those are the regions where complexity is lower in autism. So it's really a striking complementarity between a network phenomenon of connectivity and a local signal phenomenon of, in this case, entropy or complexity. Leading us to two hypotheses. One that we like, which is that if you have tight regulation by a lot of connectivity, you'll probably have low complexity because you're so regulated, you're going to only be able to do predictable things as a neuronal ensemble. If, on the other hand, you're poorly connected, poorly regulated, you can do whatever you want, and that'll be very complex. So how that might work clinically is that perhaps typically developing children have good local circuitry, good balance of GABA and glutamate, good connections, good regulation, nice coherent oscillations, whereas the children with autism perhaps have an impairment in that electrochemical balance, leading to poor phase quality of the oscillations and poor coherence. So this is basically a hypothesis that links network phenomena with local signal characteristics. There's the reverse hypothesis, which is that if you have intrinsically signals of intrinsically low complexity, it's much easier to compute some sort of measure of connectivity. So it could all be a mathematical artifact that has to be offered up as a counter hypothesis. But this one has biological sort of interest to it. So here's the same thing. If you had an intrinsically complex signal, it's pretty difficult to find another intrinsically complex signal to demonstrate a correlation with it. We prefer hypothesis 1, but you can't rule out hypothesis 2. OK, so summarizing everything, MEG has access to, and probably is, something like a 4.5-dimensional imaging sequence. It has where, it has when, and it has some spectral information as well, which is something like what is going on. And I really believe that different things are happening in different parts of the frequency spectrum, and the different correlations and coherences and connections are probably mediated at different frequencies, and there really is something interesting going on in band-limited places. I think that the metrics we use to describe this are varied, but at least for temporal metrics, we have latency, of course. We have the various moments. We could come up with standard deviation measures. We can come up with the fourth moment, the kurtosis. We can come up with entropy measures of complexity. Spectral features include at least power, phase and frequency, and I haven't talked about it, but the interaction between two things going on at the same time, so low frequency and higher frequency signals. And we can probe using these measures inter-regional relations as sort of evidence of connectivity via these measures like synchronization or coherence. And then if you capitalize, if you've got yourself into thinking about what electrophysiology offers you in terms of time and frequency, it allows us to start thinking about clinical and clinical research applications. Not least, epilepsy takes advantage of this temporal feature of spikiness, which we can image. Pre-surgical planning, that takes advantage of a spectral feature of desynchronized neural activity associated with a task. So those are actually clinically indicated benefits of electrophysiological techniques. We can also look at disorders like trauma associated with a spectral feature that is localized, pathologic, rhythmic activity in the 1 to 6 hertz range. Don't see that normally. If you hit your head, you do see it. We can look at neuropsychiatric and developmental disorders in terms of a variety of temporal and spectral features and connectivity anomalies, not least what I've shown, which is timing, latency, and quality of the gamma oscillations, or oscillopathies. And then just my usual bias, when trying to establish the basis of these markers, the biological basis, so that we can really advance our understanding from a collection of epiphenomena to a biological description, we tend to invoke information from other modalities. Might be cortical thickness, might be something about the white matter, might be something about the neurochemistry or the metabolism. And multimodality is the way to do that. So with that, I'd like to thank the various members of my lab who've contributed to this work, as well as the labs of Ragini Verma and Steve Siegel, who've done the math and mouse respectively, and the various people who've supported this work. Thank you very much.